Warm wishes to everyone. Myself, Mrs.J.Merlinrani, Assistant Professor of Nutrition and Dietetics from SFR College for Women. Today we are going to see about what is protein and the classifications of protein and its sources. So first we have to see about what is protein. So protein is a very important component for human being health, and when protein hydrolyzes, it will eat more amino acids. Each amino acids are composed by the linkage, that is peptide linkage. Individual amino acids may be considered analogs to the monosaccharides of carbohydrates. The number of amino acids in a protein molecule may range from 2 and several thousands. So protein, it means it derives from the Greek word proteus, which means of first importance. So our human body is mostly composed of protein. Each enzymes, hormones, these are all composed by proteins. So it is a major structural component or the animals and human tissues. It acts also as a biological catalyst, that is enzymes and structural parts of each organs present in human body, and it also participate in different cell reaction, and it also a major component of immunity and provide fuel also to the human body. Then the classifications of protein. So protein, it was classified as 3 types. First one is simple protein, second one is conjugated protein, and third one is derived proteins. So simple proteins, when the protein subjected for the hydrolysis, if it is yield only one molecule of amino acids, it was called a simple protein. Then the conjugated protein is when the simple proteins was composed or combined any other prosthetic group, it was called as a conjugated protein. This conjugated protein, when it was subjected for the hydrolysis process, it will yield several amino acids, that is 2 simple proteins. Then the derived proteins, when the simple and conjugated protein subjected for several reactions, this derived proteins is formed. Then these are the examples of simple proteins. The first one is albumin. So albumin is present in blood as serum albumin, and it also present in milk as lactalbumin, and the albumin is also present in egg white as ovalbumin. In lentils it was present as leguminolin. In kidney beans it was present as physolin. Then the albumin was present in wheat as leucosin. Then albumin was coagulated when it was subjected for the heat. It was found in all the plant and animal sources. Based on the sources, it was the proportion of the proportion of the albumin may vary. Then the globulin, it also is these are also the simple proteins. The globulin was present in blood as serum globulin. In muscles it was present as myosin. In potatoes it was present in tubulin. It also present in brazil nuts and hemp, and lentil also have some globulin component at leguminolin. Globulin was present in many sources. It was soluble in water and neutral solutions, that is not as acidity and alkaline. It was soluble in neutral solutions. Then it was precipitated by the dilute ammonium sulfate. While applying the heat, the globulin was coagulated. It was present in both plant sources and animal tissues. The next one is glutenins. So glutenin was present in wheat and rice. In rice it was present as vorisine. It was insoluble in water, and it was also insoluble in dilute salt solutions, but it was soluble in only dilute acids only. It was found in grains and cereals only. The next one is histones. So histone, it is a simple protein. It was present in thymus gland, pancreas and nucleoproteins. It was soluble in water, salt solution and dilute acid, but it was not soluble in ammonium hydroxide. When histones was subjected for the hydrolysis process, it was yield lysine and arginine, but in human body it was combined with nucleic acid within the cell. Next one is scleroprotein. It also a simple proteins, and the scleroprotein was present in human body in connective tissues and heart tissues. Structure is it is a fibrous structure. It was insoluble in solvents. Then it was resistant to digestion. The scleroprotein was cannot be digested by the digestive enzymes. The next one is conjugated protein. So conjugated proteins in our human body, nucleoprotein is it has a conjugated protein. So nucleoprotein, it was combined with nucleic acid and nitrogen and phosphorus. So when it was subjected for the hydrolysis process, it will eat nucleic acid, nitrogen and phosphorus. It was present in chromosomes in each cells, and it was combined with the proteins with either DNA or RNA. The next one is mucoprotein. This mucoprotein was present in mucin, that is saliva, and ova mucoid in egg white. This protein was combined with amino sugars, sugar acids and sulfates. The next one is glycoprotein. So this glycoprotein was present in bones as ooza mucoid, and tendons it was present in tendomucoid, and it was present in cartilage as chondromucoid. It was containing 4% of hexamycin and mucoproteins. If less than 4%, then it was a glycoprotein. The next one is phosphoprotein. When the phosphoric acid conjugated with the simple protein, it was called as phosphoprotein. In milk, the phosphoprotein was present in casein, and in egg yolk it was present in ova-vitelline. Phosphoric acid joined esters linkage to the protein. The next one is derived proteins. When the simple protein and conjugated protein react in some reactions, this derived proteins where we get this intestine, it was present in elastin, and myosin it was present in muscles. These are the derived proteins. When it was a hydrolysis process, it was we can get the short actions of acids and enzymes. It was insoluble in water. When the derived proteins are in a digestive process, it is an immediate product. It was called as an immediate product in protein digestion. It was soluble in water, not coagulated by the heat. So proteases was precipitated in saturated ammonium sulfate solutions. When first part of partial when the digestion, the derived proteases was get in an initial stage. It was digested by the pepsin and trypsin. So after the proteases, we can get the peptones as a secondary product. It is a smaller molecular weight than the proteases. So when comparing the proteases and peptones, proteases has the higher molecular weight, peptones has a smaller molecular weight. The next one is peptides. It is a last product. We can get the peptides in intermediate reactions. 2 or more amino acids joined by the peptide linkages in peptides. I hope this video is useful to know about the classifications of protein. We will see the next video to know the digestion of proteins and absorption. Thank you.